So the island continent of Australia has a number of different biome types, one of which is the Australian monsoon tropics, and that comprises 3 to 4 major regions, including the Kimberley, which is located at the very top of Western Australia. Now, the Kimberley region has been geographically fairly stable over the last hundreds of millions of years, and yet very comparatively little is known about the paleoclimatic history during this time. However, we do have a lot of knowledge on the current climate. It's a monsoonal climate, and one of the big things to notice is that there's quite a distinct rainfall gradient. Particularly, there's a doubling up here on the northwest coast compared to the central area of the Kimberley. And it's also a relatively geographically and topographically complex landscape. It's got an array of different habitat types. So it's been established that the Kimberley represents an area of a high endemism area for Australia. It's a biodiversity hotspot, particularly on the northwest coast, and yet relatively little is known about the interior region of the Kimberley. And this is because the area as a whole is incredibly remote. It's very difficult to access a lot of these areas, and so this has really severely limited the number of studies that have been able to be conducted in this region until fairly recently. Recent years, however, have seen a dramatic increase in the number of studies produced, indicating that there's quite high geographically structured mitochondrial diversity in a plethora of different vertebrate taxa throughout this area. But what does all of this genetic diversity actually mean?
No.
Well, my PhD is part of a larger collaborative project across the ANT, and we're looking at trying to get at the implications of this genetic diversity, and in particular answering questions regarding the effects of the landscape on the processes of diversification within this area, as well as looking at trying to assess the true level of species diversity within these regions. And we're using a number of charismatic gecko taxa to answer these questions, particularly from 3 different families, so it's quite distinct groups. And this is just some numbers to give you an idea of the type of sampling level that I'm using. To go into a bit more detail, there are 4 major distinct groups that I'm looking at in the Kimberley. First of these is the Dura, or the velvet geckos. We then have Stropurus, the spiny tails, and 2 species complexes from the group Coauthors or Tatella. And these groups are quite distinct, as I mentioned. They're broadly co-distributed, and we're gathering these really large multilocus data sets to try and answer a whole lot of questions using PopGen, phylogenetic, phylogeographic analyses, looking at comparing the different patterns of genetic diversity in the different distributions and trying to work out what's going on in this area and how these groups are diversified. And across a number of these different groups, we're seeing similar sorts of patterns coming out a number of different times, but on different scales as well. Today I'm going to mainly focus on one group though, in particular, the Adura or velvet geckos. Now, just to give you an idea, this is an ND2 mitochondrial neighbour joining tree. To give a bit of context, this blue group here represents a species complex, the closest related Adura to the Kimberley group. And these number of different species are found in pockets throughout this broad distribution. This is just giving you an idea that comparatively, the group that is represented in this tiny region in Kimberley has about the same level of diversity as this broader range here. To go into a bit more detail on Kimberley itself, there are 2 separate, distinct, and not necessarily closely related lineages within the Kimberley. The first of these is the Felicipoda lineage, containing Felicipoda proper, and introducing our newest species of Adura, which has only been formally discovered within recent months. And I'll just briefly go into a bit more detail in this group here. So the first of these, Felicipoda, we have quite limited sampling and it has a really restricted range. It's tied to this area of higher rainfall across the west coast. And yet despite this limited sampling, we're still seeing quite high diversity in this species. We then have our new species, which is currently only known from a single population found in a single mountain range at the very base of the south of the Kimberley. And this mountain range is only about 30 km long. And there's 18% mitochondrial divergence between these 2 species. Now, the reason we refer to this as possibly relictual diversity is that we are suggesting here that there's a possibility this was once a much wider spread distribution or widespread lineage, and that due to processes of climatic history changes and attenuation, these have become fragmented and separated into these remnant populations here. So these are the only areas or pockets within this region that they can survive and persist within the environment. And it's not the only pattern of molecular diversity that we're seeing. This is on a bit of a broader scale, but Pseudophagodactylus, the giant cave geckos, are also showing these patterns of refugial lineages or elliptical lineages. And this is the first sort of evidence that we're getting that maybe aerification is having an important role on this process of diversification, pushing these groups into refugial regions and then even smaller refugial zones within these regions. So just to go back and reiterate, we have our Marmorata complex, not in the Kimberley, closely related to our first lineage, Felicipota, and our new species. And then we have our second lineage within the Kimberley, Gracilis. So this group has a much broader distribution across the Kimberley. And if we break these groups really basally up, really right at the tips of the mitochondrial tree, we have a really high level of diversity, so about 15, 16 groups with about 8 to 12% divergence in the mitochondria. We can then break these clades more basally based on the structure of the tree, and that gives us about 5 to 6 different groups, and these have basal divergences of between 14 and 16 in the mitochondria, and that's quite high. So I've then taken representatives of each of these mitochondrial clades and I've sequenced them for 6 nuclear loci, run concatenations of these through Raxanella Bays. These outside groups all come out as monophyletic. They're quite distinct, which is really nice. But even though Gracilis as a whole is monophyletic, there's very little support for any of these mitochondrial lineages that we're seeing. The 2 main ones here that I'll note is this eastern group and this tiny little island off the west coast, which seems to be distinct in both mitochondrial and nuclear data. So as a really preliminary analysis, the first round of my PopGen analyses, I've run this data set through structure, and it gets between 5 and 6 groups depending on how the run goes. The first of these again is this eastern group, and this is really good because we're seeing a distinct break between the east and west Kimberley in a number of different taxa of all different types. So there's some suggestion that maybe there's some sort of phylogeographic barriers in here that have split these 2 regions. The second grouping here is along this south range. This actually follows a mountain range which bounds the southern Kimberley, so King Leopold's and Durack. And this is the first point that I'll just note here. Our groupings and structure don't necessarily follow the linkages in the mitochondrial data sets, but it's nice to see that some of these populations we're pulling out are actually making nice geographic sense. And another important thing to note about this southern range, not only do we see it in a number of other taxa, but this clade is showing much lower diversity than any of the other areas in the Kimberley. And that's sort of suggestion that maybe there's a signal of recent range expansion throughout this group or throughout this area. Then we have a north-central clade, and depending on our runs, there's some confusion as to how the groupings go in the west coast of the Kimberley, and that confusion is also seen in a lot of other groups, as that's quite a high area of mitochondrial diversity. And as I mentioned, that was just a basic preliminary analysis. We've got a number of other species limb sort of analyses we're going to be running our data sets through, as well as PopGen analyses. And I'll mention fractal diversity here. This is what we're sort of looking at in this group. The reason we're doing that is because we're seeing a number of different taxa evidence for cryptic speciation within the Kimberley zone. So things that we once thought were one species, when we're doing higher sampling and more genetic sampling, we're finding out that they're actually complexes of a whole lot of morphologically similar animals that we didn't actually notice before. So to sum up, we have a tale of 2 lineages. We have Felicipota versus Gracilis. These 2 lineages, we've run our data sets through beast analyses. They're coming out with similar times of diversification, so origins within the Miocene. And yet, despite these similar times of diversification, they're showing distinctly different patterns. And the suggestion here is that they might be responding differently to the same processes of environmental change within this environment. So within Felicipota, we're seeing a distinct pattern of retraction here from what may once have been a much broader distribution, whereas in Gracilis, we've still got this really broad distribution, and the patterns that we're seeing are more reminiscent of persistence. So possibly with this group, they've been surviving within pockets throughout the range, and maybe expanding and contracting periodically throughout time. And another link that we have is that with hypothesizing there may be differences in the ecologies between these groups that are actually causing these different patterns of diversification. So, but whilst both species are saxicoline and rocks-dwelling animals. These ones are really specialised. They're tied to specific regions within the environment that provide microhabitats that are specialised for these animals. Their morphologies are really different. They're built for climbing. And so maybe they're only able to survive in certain regions, whereas these guys, even though they are still living in rocks, they're much more terrestrial. Their body shape's flattened. They're seemingly a lot hardier. We're finding them in really scrappy sort of rocky environments throughout the area. And so maybe that's allowed them to persist and survive through climate change that the other ones haven't been able to survive in. And that could be allowing some more higher levels of diversity and dispersal throughout the environment. Now, the two key take-home messages I'd like you to take from this talk is that whilst these ritual areas are more noticeable in the Felicipota lineage, they're still significant for both of the taxa that we're seeing here. So particularly this west coast region, which Felicipota is only ever found in, it's the pocket that is holding this species in one lineage, but in the other it's also an area of hyperdiversity. It's the area that we find the highest number of mitochondrial lineages, and we're seeing that in a number of other taxa that we're looking at as well. Similarly, in the southern range, that's the only pocket that this new species is found in, and in Gracilis, it represents a possible area of range expansion throughout that zone. And that's, again, another pattern that we're seeing in many other taxa. And finally, it would seem that it's actually a complex interaction between the landscape within this region, so different types of mountain ranges here, and also this really different climatic history within the region, and also the difference in the rainfall that we predict might be driving differences in these patterns of diversification within these different groups, and possibly linking to the ecologies of the animals themselves. And I'd just like to thank everyone who's made this project possible by funding, helping out getting sampling, doing field work, and my supervisors, and also for the funding that's provided me to come over to this.